All right. Well, thank you very much. And hi, everybody, from your home, clinics, campus, and so on. It's a very big pleasure to be here today and to talk to you about the latest advances in this wonderful field. I am not a dentist, but I'm an oral microbiologist, and I fell in love with this field several years ago now, like 15 years ago, when I tried to apply high throughput sequencing and metagenomic techniques to study oral microbes. And I think we are in the middle of a very big change in the field. We are in the middle of a revolution in the field of oral microbiology that I think we are going to sense very soon in terms of the diagnostics, in terms of the understanding of oral diseases, in terms of the new preventive measures that we can apply to our patients. And sometimes, when you are in the middle of a revolution, it's very difficult to see it. But I'm pretty sure that when a few years pass and we will look back, we will see that we actually are having very profound changes, in some cases motivated by the arrival of new techniques in the field that have allowed us to have a novel and more complete view of what is going on in the oral cavity. So I have divided the talk in several blocks, and I'm going to make a summary about the important features, in my opinion, about each of these blocks and how they influence the field, with a particular emphasis on the clinical implications of the work. So we talk about the novel techniques. It has been very interesting, the application of this new classification method in microscopy, by which you can actually visualize different species of bacteria all at once. In the past, we could do this with the fish method, the traditional fish method, where we could label one or two species, three if you were very good at it. No more than that, because then the wavelength of different of the markers would overlap, and then you would not be able to distinguish them. But now, by the combination of several of these fluorophores and then some bioinformatic analysis, we can actually label tens of different bacteria, each of them with a different probe, with a different fluorescent marker. And as you can see here, this is an image of the tongue biofilm, and each bacterium here, each bacterial species is labeled with a different color. So one of the things that you can actually see colonies, like real colonies of bacteria, like streptococci here. And then you can see that some bacteria appear always to be close to each other. So like these yellow ones next to streptococci. So it seems, for example, that we have some bacteria that produce lactic acid, and they are normally close to other bacteria that actually use lactic acid as a carbon source. So we are starting to see why they get organized in a particular way and how these social networks of bacteria work. So this is an image of the tongue, and I will show you some others later about how the biofilm in dental plaque is organized. The second set of techniques that have been instrumental for this new period in the field is actually the arrival of the high throughput sequencing techniques. In the last 10 years, we have mainly used second generation sequencing, like Pyro sequencing and Illumina sequencing. And now we have the arrival of the third generation sequencing techniques, which are actually single molecule techniques that are unbiased. You don't need to do PCR. And some of them, like the nanopore sequencing, are just like magic. You have the DNA that actually goes through very small pores that, and then you can measure an electrical field that changes according to whether you have an A, a T, a G, or a C. And that is done at a really high speed. And you can see in real time how the sequence is being obtained. The same for FISABIO sequencing. I think it's also a big step in the length of the sequences, because we could obtain 400 or 300 base pair long reads before, and now with FISABIO and nanopores, we have length, reads sequences which are several thousands of base pairs in length, and this is really going to be it's already being a revolution in metagenomics and for doing genomic assembly and so on. We have also improved a lot the techniques to measure oral biofilms in vitro, like different in vitro models that try to mimic what actually happens in the real life in the oral cavity. This is a technique, for example, where we can measure the biofilm growth in real time. And this is by the use of impedance, which is the measurement of an electric current that goes through the culture medium. So what we have here on the right is we have 96 well plates. And at the bottom of the wells here, we have gold electrodes. And then we pass an electrical current through that. If there are no bacteria, or if the bacteria do not form a biofilm and they are actually, they grow planktonically in the culture media, then that is not going to impede the electron flow. And then we will have, you know, a standard, a normal electrical current. But if we have the bacteria that actually physically attach to the bottom, forming the biofilm, that is going to impede the pass of the electrical current. And that impedance, that electrical impedance, can be measured in real time. And the higher the impedance to the current, the thicker the biofilm is. So we can actually measure how the biofilm is growing in real time. It has multiple applications because we can study the dynamics of biofilm growth, but we can also look at how oral biofilms are sensitive to different antibiotics or antiseptics, as this curve shows, growth curves show here. So we have here the amount of biofilm on the y-axis, and on the x-axis we have the time. So we have here, for example, this is the control. We see here how in a few hours the biofilm grows. In this particular case, we are taking real samples from the periodontal pockets. In other occasions, we take samples from the root canal in endodontic infections, and then we take them ex vivo, we take it fresh into the lab. We grow them here, and then we can see how the biofilm grows in the absence or in the presence of different antibiotics. So here, for this particular individual, this sample, we see that this antibiotic is the one that works the best. So this should be the right antibiotic for this person. However, we have here another case on the lower left where none of the 3 antibiotics that we have tested actually work. So, you know, the biofilm growth is the same. Here, this is interesting because we have a case where this antibiotic is actually one that works the best. So this is different from the other one, you know. This really shows that the antibiotic susceptibility is patient dependent. It really varies on every single sample that we take. And actually this antibiotic, which is marked in blue, which appeared to be the best one in this particular patient, well, for this sample, not only doesn't do anything, but it actually induces the growth of more biofilm. So the bacteria and the biofilm appear to be resistant to that antibiotic, but they still feel that antibiotic, they sense it as a stress. So what they do is to produce more exopolysaccharide, more biofilm to protect themselves, and therefore there is an induction. So that would be a really bad antibiotic to give to this particular patient. And then we have here on the top left another case where all 3 antibiotics that we have tested actually induce more biofilm formation. So if we are going to give this antibiotic to this person, it's going to not only not do anything, but actually increase probably the inflammation in the gums because this subgingival plaque is actually resistant to these antibiotics, and we would need to test someone else, some other antibiotic. So this really, I think, in my opinion, opens the way to, it paves the way for personalized dentistry in the selection of the right antibiotic to each person. I think the biofilms are different in different individuals, and one antibiotic doesn't fit us all. So I think it would be ideal to maximize the efficiency of antibiotics and to make a more rational use of antibiotics, and also to reduce the level of antibiotic resistance in the population, to actually select the right antibiotic for each person. All right, the second block I wanted to talk to you about is actually the structure and formation of the biofilm. And I think this is very important because, as you know, in all textbooks, we always say that oral diseases like dental caries, gingivitis, gum diseases, and so on are biofilm-mediated diseases. What does it mean? That the more biofilm we have, the more chances to develop the disease. A biofilm, as you know, is an accumulation of bacteria that attach to a solid surface like the teeth, in combination with different glycoproteins of the saliva, remnants of buccal cells and so on. They produce this extra polysaccharide matrix that allow them to grow and to protect themselves from external environment and environmental pressures. If you go to textbooks, this is the standard hypothesis about how oral biofilms, dental plaque, is actually formed. It appears that some bacteria appear to be early colonizers of the biofilm, so they are able to attach specifically to the acquired pellicle formed by glycoproteins in the saliva. And then we have a beautiful succession where, this is the theory, a beautiful succession of attachment of other organisms, because some of them possess specific receptors where other bacteria that have a specific adhesins are going to be able to attach and to co-aggregate. In this photo on the right, we have one of these cases of co-aggregation between Fusobacterium and the main gingival pathogen Porphyromonas gingivalis. So this is supposed to happen at this early colonization step, and then we have the arrival of Fusobacterium nucleatum, which is this very long fusiform shaped bacterium that appears to be a bridge between early colonizers and late colonizers. Actually, by looking at this, if we had to target a particular bacterium to prevent the formation of a mature biofilm, I would probably target Fusobacterium, because there are really many pathogens like the red complex species, involved in periodontal disease that actually attach to Fusobacterium. Fusobacterium is the species, the known bacterium that has the largest number of co-aggregation with many other bacteria. It's a nasty organism. It has been involved in colon cancer. And therefore, I think if I had to develop a vaccine against some oral bacteria, this is the one I would choose. Okay, so this is what we all have in our mind about how dental plaque is being formed. But there are a few things that actually don't fit with what we know. Look at, for example, the position of streptococci. They're supposed to be early colonizers, so they should be here close to the teeth. However, when we have these classified images, it actually is the other way around. So this is an image of a 24-hour dental plaque. So this is what your oral biofilm on your teeth surface looks like if you don't brush your teeth for 24 hours. And the part here on the left is the part of the biofilm which is attached to the teeth, and the part on the right is what is exposed to the oral cavity. And the first thing that really strikes us is that the whole biofilm architecture is dominated by this bacterium, Corynebacterium matriciotti, for which we know very little about, that forms these very long filaments from the base to the outside in a radial way, like a hedgehog structure, as the authors called it. And then, it is like the keystone organism in the biofilm architecture. And then at the edges of the filaments, here labeled in green, we have streptococci. So they are actually outside, not close to the bottom, but actually on the opposite side from what the current model of biofilm formation dictates. So it seems that they are on the outside because they are able to live in the presence of oxygen, and therefore they form like a shield, to protect the inside of the biofilm, allowing the anaerobic atmosphere to be formed in here. And this will allow strict anaerobes like Fusobacterium to actually grow, which is marked here in white. So it is true that Fusobacterium appears to be in the middle, but actually streptococci are not at the bottom. They are actually on the outside. So there is something in the model that doesn't fit. The second part of the model that doesn't fit is that we don't see planktonic bacteria in saliva that are there floating, looking for new sites to co-colonize. So in all textbooks, this is the image that we will have about how a biofilm is going to be formed. So we have individual planktonic organisms floating in the media. In our case, they should be floating in saliva. Then they would attach to the surface, and those individual bacteria will start to grow. They will produce these exopolysaccharides, forming the matrix. And then the biofilm will grow to the point where there are no more nutrients, and then they will detach from the biofilm and again go into the saliva to look for new attachment sites. Okay, so then what we do was to look for these bacteria in the saliva. And actually, after when we first looked at this, we realized that bacteria are never planktonic, or they are hardly planktonic. They are, most of the time, they are forming these aggregates with many other bacterial species, or because they attach to buccal epithelial cells that detach from the oral cavity. And when we saw this, we just couldn't believe that that bacteria were not planktonically like we had learned from textbooks. So we did this by many different techniques. We sampled all the saliva of the people in the department, and we always found the same thing by different methods. So here we have this aggregate by TEM, by SEM. Here we see how they attach to the buccal epithelial cells. And it's very interesting because then what we did was to quantify and to count the number of bacteria which are individual and the ones which are forming part of aggregates using flow cytometry. And actually over 95% of the bacteria appear to be forming aggregates. So again, this image that we have here is not true either. We see actually that we have aggregates that should attach to the surface. And is this important? What happens then? How does the biofilm grow? Well, when we use this bioflux system, which is a system where a microfluidic system where we can have the saliva passing through the system to which, to a microscope to which we have applied there a camera, we can see how this moves. And you're going to see that the bacteria which are alone do not grow, whereas the bacteria which are forming part of an aggregate are the ones that actually form most of the biofilm. We can also see this in 3D by using confocal microscopy, where we can reconstruct the 3D structure, and we find the same here. So in these two arrows are marking bacteria which are isolated, which are individually attached to the surface, and then we have here aggregates that have also attached. And as you're going to see, the bacteria which are alone, they attach, but they are not able to grow, whereas the biofilm actually starts from these aggregations, which actually act as nucleating agents, for the biofilm to grow. So you need the bacteria to be forming these multi-species consortia in order to form the biofilm. So now we think actually that the structure, how the biofilm is formed, is different. We don't have early and late co-colonizers. We have all the bacteria are there from the beginning. And what happens is that some bacteria are able to grow early, for example, because they can grow in the presence of oxygen. And then they change the atmosphere, they change the environment, for example, because they consume the oxygen, allowing anaerobic organisms to grow, or because they are able to produce lactic acid that can be used by other bacteria. So this is really changing our view. And I think one of the problems here is general in the field of microbiology at large, because we have been extremely biased by the pure culture. I think from the times of Louis Pasteur and so on, we are used to working with bacteria in the lab alone in a pure culture, which is very convenient because then we don't have to deal with the heterogeneity of bacterial populations. But at the same time, when bacteria are growing on their own in the lab, they don't behave the same as when they are forming part of a bacterial multi-species structure. So I think we need to find new ways to study bacteria in these complex interactions. For example, by doing co-cultures, by doing metagenomics or metatranscriptomics, where we can actually look at all the transcriptional profile of the whole community of organisms, and in order to understand much better these interactions, because I think they are instrumental as we are seeing about how the biofilm is being formed. We need these multi-species, these bacterial consortia to be present to actually form a biofilm. And it's very interesting that these multi-species structures actually go beyond kingdoms. So you can have interactions and co-aggregations between bacteria and fungi, like this one that we find here between Candida albicans, a yeast, and Staphylococcus mutans, one of the bacteria causing dental caries. And actually, when they are together, they change entirely the properties of these two species. So when they're together, it seems that they produce more acid than when they are growing individually. And also they appear to be more resistant to antibiotics and antiseptics than when they are on their own. Not only that, but actually they can walk and they can move together. And actually the group of
Michelle Koo in Philadelphia have even estimated the speed at which they do that is about 40 micro per hour, you know. So it is quite interesting, and that's why they have called this structure supraorganisms, because they actually change the biological features of them individually. You can see how they move in this video. I'm going to show you here on the left. So here we mark streptococci, here we mark the candida, and if you look at the top panel, we have labeled both, you know, the yeast and the bacteria, and you will see that with time, the yeast start to produce hyphae, and then as if they were legs, you know, they carry on their shoulders the streptococci, which at the same time they also grow. I'm going to put it again. So these green are the streptococci. They grow while they are being transported by these little jumps of candida by using hyphae from one place to another. So I think we are now at the beginning of understanding these multi-species interactions that go beyond bacteria, and they can be between bacteria and fungi as well. So I think this is this is already changing our way of understanding how oral biofilms work. Okay, I have to talk a little bit about salivary diagnostics. I think this is a very important field in dentistry, and I think it's going to be, we are going to see more and more of this also in the dental clinic. Of course, we can study, for example, different biomarkers in the gingival fluid, and those biomarkers could be indicative of the person having inflammation and having gingivitis or periodontal disease. And the interesting thing is that many of these biomarkers in the gingival fluid, which is the ideal fluid to actually examine, you know, or to evaluate gingival diseases, because we are taking the sample from the site where the disease takes place, many of these biomarkers appear to correlate with the concentrations that you find in saliva and with similar levels of sensitivity and specificity. So this is a systematic review and meta-analysis where we see that the values are similar when you consider especially IL-1 beta, MMP8, and IL-6. These three appear to be very good biomarkers in saliva of periodontal disease. So this means that, you know, with an easier to collect sample like saliva, you can actually measure there some particular biomarkers which are going to tell you whether that person has the disease or not, and the degree of the disease. But I want to take it further, because not only we have biomarkers of oral diseases in saliva. Saliva is a wonderful fluid that actually captures in real time the general health state of our body. We produce more than 1 liter of saliva per day under normal conditions, and we are all the time swallowing this saliva and producing new saliva. So in real time, the composition of saliva is changing and is going to give us a general overview about what is going on in our body. In some cases, for example, if we have a cancer, some small RNAs are being produced and are being released from cancer cells when they die, and they end up in saliva. So the group of Professor Wang in California, for example, has developed a method to actually quantify the presence of these small RNAs in saliva, and they appear to be very sensitive to detect different cancer types and other general systemic conditions. This means that, you know, in the future, we can actually have a saliva test to detect many systemic conditions without the need for a blood test or without the need of taking a biopsy to detect a specific kind of cancers and other diseases. This is just to give you an overview the different biomarkers of different cancer types that you can detect in saliva. And these, you know, new ones come every year. So a few months ago, for example, it was found that the biomarker that is normally used for for prostate cancer in males appear to be even more sensitive when you measure the levels in saliva. So I think we are going to see more of this. And in the future, the way I see it is that the dental clinic could be the perfect place to actually monitor health and to have early diagnostic tests when a person goes for a dental checkout or to have a filling or something. Because the thing is that when we normally go to the doctor, to the medical doctor when we are ill, when we are sick, but we actually go to the dentist when we are healthy, when we are systemically healthy. You may need, you know, a filling because you have a cavity, or you just may go for a checkup, you know, for a yearly checkup and so on, which means that you don't have to be ill. And therefore, I think it's a perfect time for donating a saliva sample, and we can actually, you know, perform a test for systemic diseases there. And then it will be a very wonderful way to have a window of opportunity to identify the disease before the clinical symptoms appear. And I think in the future, you know, if all the dentists and the people working in the field are clever, I think we might be able to develop these experimentally, and also to apply this test in the dental clinic. Actually, I want to mention that not only saliva is becoming a very interesting diagnostic fluid, you know, the new liquid biopsy, but even the breath. This is an example from a company in the US, Breath Diagnostics, that have developed a system by which you can actually, by just expelling your breath, you can identify several molecules, several volatile compounds that appear to be very sensitive and very specific or particular types of diseases. In this case, these 4 compounds here appear to be at significantly higher levels in people with lung cancer. So you can identify lung cancer simply by a breath of air. And they are very sensitive. So in parts per million, even in parts per billion, you know, the levels of some volatile compounds that normally are not present in our breath appear only under certain disease conditions. So I think this is also, there are more cases of people working on these for the identification of type 2 and type 1, that the diabetes and many other diseases. I think we will see more of this in the future. All right, and let's move to another block, microbial etiology of oral diseases. So I'm going to look a little bit at the microbial etiology, you know, which organisms appear to be behind different oral diseases. Okay, we have done a lot of work on this, and by doing DNA and RNA work on cavities, RNA is the active part of the genetic material, so it's not only dead bacteria that we can identify, by bacteria which are actively growing in the sample. And this is the bacterial composition in different dentin cavities from different individuals. Each color represents a particular bacterial genus, and as you can see, the composition of bacteria is very variable in cavities from different individuals. So the first thing is that we have many bacteria which are active, present there. So it's not a typical infectious disease where you only have one particular pathogen responsible for the disease. We have many of them, actually. We have tens of them. And actually, not only it's a clearly polymicrobial disease, but also it varies between individuals, making it very difficult to have a single target that we should aim at if we wanted to control this disease. This is the most extreme case that we have found, because not only different individuals have different bacterial compositions in their carious lesions, but even within the same individual it varies, and even within the same tooth. So this is a molar, this is a 4-7 with 2 different cavities, and we can see actually that the composition is different between them. Here we have this organism which is not present here. Here the most common Streptococcus is Streptococcus from the mitis group, and here this is Streptococcus sanguinis. And okay, where is Streptococcus mutans, you know, which is considered to be the causing agent of dental caries? But it's only this very thin line here and in here, which represents about 1% of the total. That means that even if we can actually have a vaccine against dental caries, where we have used the surface antigens of S mutants as the target for our vaccine, even if we are able to produce antibodies against that organism, the other 99% of bacteria which are able to produce acid are going to be alive, and they will be able to keep expanding the cavity. Therefore, I think this is the reason why a vaccine against dental caries is going to be very difficult. And for decades, there have been many research teams trying to develop both passive and active immunization strategies. And then when they go to clinical trials, then it doesn't work. And I think this is the reason. It's a clearly polymicrobial disease. It is not a traditional, infectious disease where we have one pathogen causing it, and therefore we cannot treat it as an infectious disease. We cannot try to develop a vaccine. It is not going to work. And this is not only true for dental caries, but also for example, gum diseases. So here we have the composition of bacteria in different individuals that vary in the depth of the periodontal pocket. So if we move here to the right, deeper pockets, more advanced stage of the disease, we see an increase in some bacteria which appear to be responsible of this inflammation and immune alterations and so on. So we have here the typical red complex species of gum disease, but I want you to look not at what happens under disease, but what happens under healthy conditions. So what happens when the depth of the pockets is below 2.5 millimeters? So we don't really have a pocket, we have a healthy gingival sulcus. Okay, we take these lines from the pathogens down here, we go back, okay, we are here. So these are healthy individuals. And as you can see, it's not that healthy individuals don't have this bacteria. They do have it. I always say that we all have the enemy at home. We have all these periodontal pathogens in healthy individuals. It's just that they are below 1%. So they are very low levels. It seems that they are kept under control. There is an homeostasis. There is some balance between the healthy bacteria, which account for most of the ecosystem, and these pathogens. So those pathogens are present. It's not that we get infected with them. Imagine that if you eat some particular item of food that contains salmonella, you are going to get infected with salmonella and develop salmonellosis. So you are going to develop a disease because you get infected with a bacterium that you didn't have before. Or the same, if you drink a glass of water with Vibrio cholera, you are going to get infected with that organism. Oral diseases are something different. They are not infectious diseases. They are the product of a dysbiosis, which means the end of the breakdown of the balance between the host and the bacteria, because there is some external disease driver. For example, the diet, high in sugars, for example, stress, for example, a lack of or a bad oral hygiene, which is going to favor some bacteria. And then these pathogens that appear to be at very low proportions, then they increase in proportion, and then there is a disbalance, there's a dysbiosis. And that's what is causing the disease. So if it is not an infectious disease, we cannot treat it as an infectious disease. If it is a dysbiosis, then what we have to do is try to restore that balance. And that's why I believe so much in the use of prebiotics and probiotics, because they really try to restore that balance by increasing the proportion of health-associated bacteria and decreasing the proportion of pathogenic organisms. I think that we have to aim at that. And this image, I think it illustrates this very well. So in the traditional external pathogen diseases, you don't get the pathogen, you don't have the pathogen in your resident microbiota, and then you get infected by this external pathogen that is able to colonize. However, in oral diseases, where we already have the pathogens, we have the enemy at home, and then there is an external driver of the disease, like a bad diet or bad oral hygiene, which favors the growth of these pathobionts. So this is why we call them pathobionts and not pathogens, because they are already present under healthy conditions, and we need to restore that balance going from here to here. How can we do that? Because this dysbiosis is the basis for dental caries, gum disease, and also halitosis, which is also a lack of this balance, this dysbiosis process on the tongue surface. Okay, so what are prebiotics and probiotics? This is another block I wanted to talk to you about. Probiotics would be, live organisms which are health associated, and when we provide them at sufficient numbers, they can provide the benefit for our health. And we can also provide oral prebiotics, which is actually, they work as a natural fertilizer, because what they are, prebiotics are nutrients or compounds that when we add them to the oral cavity, they favor the growth of healthy organisms. So they benefit, they promote the growth and the beneficial activity of these health-associated bacteria. So I'm going to put you some examples. In the case of prebiotics, probably the most famous one is arginine, which is already in the market in toothpaste. And what happens is that arginine is used by some health-associated bacteria to be transformed into ammonia. And ammonia is a base that is going to buffer the acidic pH. So when you have a very acidic pH, as in a cavity, then the good bacteria are going to transform arginine, if it is available, into ammonia, and that ammonia buffers the pH, and therefore you prevent caries. And probably because of that, caries-active individuals have a lower arginolytic activity. The capacity to transform arginine into ammonia is lower in these individuals, whereas caries-free individuals that never had a cavity in their life, they have higher levels of these bacteria, higher activity, enzymatic activity to transform arginine into ammonia. And the good news is that if you had caries in the past, but you treat them, so you have fillings, then the level of this arginolytic activity is also high, is also as high as in caries-free individuals. So that's why arginine is present in toothpaste. And there are other examples which are being explored at the moment. So this is some work from a Belgian group a few years ago, where they actually tested different compounds to see whether they actually had an effect in the reduction of pathogens and increasing the proportion of health-associated bacteria. So I think there are, more work being performed about that. My favorite case is actually nitrate, which we have worked quite a lot. Nitrate, NO3, is an inorganic compound which is present a lot in fruits and mainly vegetables. So it is found at very high levels in green leafy vegetables like spinach, lettuce, celery, rucola, and so on. It's also very common in beets, like in beetroot and so on. And what happens is that we humans don't have the enzymes to transform nitrate. So we will get rid of nitrate in the urine, and we are not able to do anything with it. It's a very stable molecule. But oral bacteria, only oral bacteria are able to, some oral bacteria, health-associated bacteria, are able to transform nitrate into nitrite, NO3 into NO2. So they're able to reduce the nitrate and use it. And this is very beneficial for oral health. So this is what happens after 5 hours when an oral biofilm is exposed to nitrate. Most of the nitrate is gone because they transform it into nitrite. By 9 hours, all the nitrate is gone. And then it transforms into nitrite, NO2. And by 9 hours, the nitrite is also gone. So they transform the nitrite into something else. And surprise, surprise, they produce ammonia as well. So it works in a similar way as arginine. But in addition to that, it's very interesting because the enzymes that reduce nitrate into nitrite and ammonia, they consume lactate because they use lactates as an electron donor. And this is very interesting because it means that not only you buffer the pH by high levels of ammonia, we see here that in the presence of nitrate, ammonia levels are higher than in the control, but also lower levels of lactic acid. And this is because lactate is being used by the bacteria, by the enzymes, by doing this reaction. So it produces lower acidogenicity, and it also buffers the pH. And as a consequence of that, the pH in the control goes down, but in the presence of nitrate, the pH does not significantly change. So this is why eating vegetables is actually good for your health, because it prevents dental caries, because it prevents acidification. But you have to notice here also that nitric oxide is produced. So this nitrite can be transformed either into ammonia or into nitric oxide. And many oral pathogens, especially strict anaerobes, like those involved in halitosis and in gingival inflammation, they are strict anaerobes, and they are very sensitive to nitric oxide. So this means also that by eating vegetables rich in nitrate, we can also prevent halitosis and gum diseases, as we have shown already in vitro and also in clinical studies. In addition to using the nitrate as a prebiotic, we are also isolating these nitrate-utilizing bacteria as probiotics. And we have developed this method to actually find them in the oral cavity, because nitrite, you can use a particular dye, which is red, it's called the Gries reagent, and it binds to nitrite. So we put nitrate in the agar plates, and then we put this reagent that turns red in the presence of nitrite. So the colonies of bacteria which are able to transform the nitrate into nitrite appear to be red. And by doing this, we have been able to find these bacteria in oral samples, and we have selected the best bacteria that have a very efficient nitrate reducing capacity to produce nitric oxide, ammonia, and so on. Okay, what other health-associated bacteria can we find in addition to these bacteria which are able to use arginine or which are able to use nitrate? Well, in order to do that, what we did in the past was to look at which bacteria appear to be at high levels in caries-free individuals. So what we did was the first metagenome, the first DNA analysis of dental plaque that was published. We collected dental plaque samples from caries-free and caries-active individuals. We extracted the total DNA of dental plaque, we fragmented, and then we do high throughput sequencing. So we can look at which bacteria are present in here. Even if we cannot culture, we can see still their DNA. And then what are they doing? And it's very interesting because we found some bacteria associated with good health. These two, Neisseria and Rothia, we didn't know at that time why they were so common in healthy individuals. And now we know, because these two bacteria are the ones that mainly use the nitrate. So now it has been very beautiful to be able to close that circle and to understand why, more than 10 years ago, we found Rothia and Neisseria at high levels in caries-free individuals. They are also at high levels in individuals without gum disease, in halitosis-free individuals. So they're always associated with health. And it is because they are able to use the nitrate from the diet. At that point, at that moment, we were more struck actually by the presence of what appeared to be a new species of Streptococcus that was not present in databases, and it was at high levels in caries-free individuals. So we went back to them and we managed to culture it and isolate it. We did all the biochemical testing and genome sequencing and so on, phylogenetics, and we described it as a new species that we call Streptococcus dentisani. It was, for me, very exciting, to be able to name a new species for science. It is this beautiful Streptococcus that appeared to be at high levels in caries-free individuals. So we thought, okay, perhaps it's able to outcompete S mutants and all the other caries pathogens because it occupies the same niche, it grows faster, who knows. So we did experiment actually where we had a lone culture of S mutants, S sobrinus here, some of the caries pathogens, and then we put in the middle these bacteria that we isolated. And as you can see, there is an inhibition ring around. You can see here, on the left, we have Streptococcus dentisani in the middle, and then around this dentisani, S mutants cannot grow. It was extremely exciting at the moment. It was one of the most exciting days in my life when we actually realized that caries-free individuals have high levels of bacteria which are going to form as a shield. They are going to protect us from the pathogens because they kill them. So they produce antimicrobial peptides that are going to diffuse in the agar, preventing the growth of bacteria. And that's exactly what we think is happening in the oral cavity of caries-free individuals. So we wanted to know what it is that they are producing. And then we saw that they produce 14 antimicrobial peptides. These in bacteria are called bacteriocins. And from these 14, 9 of them are totally unique. We have called them denticins, from denticin A to denticin H. And they appear to be antimicrobial peptides that bind to the cell wall of pathogens and they kill them. So most of these genes encoding the bacteriocins in S denticin appear to be all close to each other in the genome in a cluster. There are these very tiny genes here because they encode peptides which are between 30 and 50 amino acids, so they are very small. And in this cluster of the genome that we have called anti-caries defense cluster, ACDC, because I liked a lot this music band in the past, and I thought it would be fun to give it that name. So now we are studying each of these denticins to see against which oral pathogens they are active. And we have done several clinical studies showing that it actually works. It buffers the pH, it lowers gingival inflammation, and so on. The way that we apply it in the clinical studies, we had the dentisani in a powder, then we put it in a dental ferrule, similar to the ones that you use for whitening your teeth, you know. We have the bucco adhesive gel with the Dentistry there. We apply it for 5 minutes, and then we have tried to put it only once, one day, that didn't work so well. Once per day for a week, which worked much better, and then every other day for a month, which is the one that worked the best. And yeah, so we are very close to take this into the market. So it's very likely that the Dentistry is going to come into the market in Europe by the end of the year. So this is a very exciting moment where something that we have discovered in the lab is going to be applied to improve people's health. And that's the most exciting thing, the best prize that any scientist can have, I think. All right, in this block, I'm going to talk about some of the strategies, some of the old ones that for which we have new information, and some new strategies, like these nanoparticles, for example, that we have developed with chemists here at the university, that contain nanodrill, which is a physin, which is an enzyme able to degrade, you know, to drill on the matrix of the biofilm. And then it contains inside these porous nanoparticles, it has an antiseptic that is going to be released only when the pH is acidic. So they are intelligent nanoparticles that when they hit the biofilm, if it is acidic, then it opens the gates, the molecular gates, releasing the content. In addition, these chemists have been able to put a nano engine, you know, that is able to propel the nanoparticles so they can move, making holes in the biofilm. We have seen that it works very well disinfecting the root canals in endodontic infections. And we have demonstrated this ex vivo in extracted teeth, and we are now fighting to, you know, all the regulatory issues to be able to apply it in clinical trials. This is very interesting because most people think that fluoride is very important from a chemical point of view because it creates this fluorapatite shield, you know, that is going to protect the enamel because it's going to make it more resistant to the acidic shock, and that's true. But in addition to the chemical effect, we have seen that fluoride also has a microbial effect. It's not that it kills bacteria, but it actually inhibits the enzymes that produce acid. So when we did the metatranscriptome, you know, the genes which appear to be active or inactive in the presence of fluoride, we see that most of the enzymes that appear to be inhibited in the presence of fluoride are involved in sugar fermentation. And as a consequence of that, they produce less acid. And this is what happens when the oral biofilm is exposed to fluoride. The pH drop is much lower compared to the control that goes down a lot, you know, to pH 5 in here in the experimental conditions where we tested. When we have fluoride, the bacteria do not die, but their enzymes, fermenting sugars, are inhibited, and as a consequence of that, less acid is produced and the pH doesn't go down so much. So I think that, you know, even the mechanical removal of the biofilm will be very important, but if we also have their toothpaste with fluoride, then not only we are going to have a chemical effect, but also an antimicrobial effect in the sense of having a lower acidogenicity of the plaque. And there are also new methods to combat biofilms, which are actually unrelated to antimicrobials. So they are not antibiotics. And one of them is actually being able to block the quorum sensing signals. Quorum sensing is the way that bacteria communicate with each other. So they produce these little molecules, which are called quorum sensing, and they produce the molecules, and they also have receptors for that. What happens is that when you have many bacteria in a population, the concentration of these quorum sensing molecules is high enough that they are going to sense each other. So it's a way to know how many of your companions are out there. So when the levels of these quorum sensing molecules are high enough, then the bacteria all decide to do something together. For example, they decide to produce the biofilm, you know, to produce exopolysaccharides, or they decide to produce a toxin. This is very common in nature in pathogens. So you don't want to, you know, to start a war if you are not enough people. So what they do is to use these signals to communicate with each other to know how many of them there are. So in a way, if they are going to form the biofilm based on these signals, if we could actually block these signals by quorum quenching molecules, this would be perhaps an alternative to having antibiotics. This is exactly what Ana Otero at the University of Santiago de Compostela, the north of Spain, has done. Her team actually found a quorum quenching molecules, an inhibitor of these communication signals produced by a marine organism, actually. And what happens is that when you put that in oral biofilms, both from healthy individuals or individuals with gingivitis or with gum disease, you see a decrease in the biofilm formation in basically all cases. So you have a decrease, between 10 and close to 100% of the biofilm size reduction. This is spectacular, and this is not killing the bacteria. This is just inhibiting the way that they are able to communicate with each other. I think this is a very clever way, and I think this is something that we should do more research in the future. Actually, this quorum sensing inhibitor was isolated from a marine organism, from bacteria from the sea, but we have identified that many of these quorum sensing inhibitors are present in the oral cavity. So I think those are probably going to work much better, and we should do more research about it. And I think this is the last block I'm going to talk to you about today. And the last years are really been absolutely spectacular in understanding what are the systemic effects of oral microbes. Some of these are good and others are bad. I think we are more familiar with the bad effects of oral pathogens. This is the traditional image that was, it's present in many books. This is what we knew like 10 years ago. We know that especially in people with gum disease, when you have all these periodontal pathogens, they are going to produce inflammatory molecules, or parts of the bacteria are going to be able to go into the bloodstream, and therefore they are going to create inflammation systemic effect, which affects many different aspects of our health. For example, an increase in cardiovascular disease, in cardiovascular risk, or even having an increased in preterm birth and pregnancy complications. It's very interesting because there have been several very good clinical studies in Brazil, actually, where when the pregnant mothers that have bad gingival health are treated of that during the pregnancy, the rates of preterm births actually go back to normal. So this is very interesting because it can be reversed. This is what we knew, but in the last years we have actually learned that some Fusobacterium, an oral bacteria, can translocate from the oral cavity to the gut, and when they go there, they can be involved in colorectal cancer. This is an example of a mouse model of colon cancer that was treated with antibiotics. The antibiotics actually kill this Fusobacterium, and the progression of the tumors was stopped. Not only that, but also 20% of the tumors that were already present, they disappeared after antibiotic treatment. So this has opened a whole new field about studying what's the role of these oral bacteria in colon cancer. And not only they are able to transform healthy cells into tumor cells, but actually they appear to be instrumental for the migration of the tumor cells to other parts of the body. So here what we have on the left is a culture of a tumor in the lab without the Fusobacterium. So this is the control. And in the right, I'm going to show you a video. In the right, we have this tumor, this colon cancer tumor that has been cultured with the Fusobacterium, this oral organism, which is marked here in this pink color. What happens, you'll see that the Fusobacterium is able to invade the tumor cells, and then it activates a particular pathway that will make the cells to migrate. When you put the video, and you will see how on the right, the cells start to migrate. So you have the bacteria there, the oral bacteria going to the gut, getting inside the cells, as you can see here. So they become intracellular, they activate the genes, they take the control of the cell, and then they activate the migration. So we have oral bacteria inducing a metastasis process. And this is why when they sent us samples from the liver in people that had already a liver metastases that we actually detected Fusobacterium also in the liver, because they travel inside the tumor cells from the colon to the liver. So I think it's just unbelievable that bacteria can have those effects, and I think we need to explore these more and to include, periodontal treatments as part of the planning of the protocols in people that are first discovered with colon cancer. I'm going to pass this slide. This is just to show you that when we have done the metatranscriptome, the active bacteria in the tumors, the ones which appear to be at higher level of activity, most of them are oral bacteria. So it's not only Fusobacterium that migrates from the oral cavity to the gut, but also these other bacteria. And I think they form these consortia, which we believe are instrumental to be able to travel from one place to another, to protect each other. And because we are seeing that they have some metabolic complementation that actually allows them to grow better when they are, with each other. But not only oral bacteria appear to be involved in colon cancer, but probably also in other diseases with difficult etiology. So recently, in post-mortem brains of people that died that had Alzheimer's, some researchers have identified gingipine. Gingipine is the main toxin produced by Porphyromonas gingivalis. Here, this is a staining of gingipine in the gingiva. So you can see in a person with gingivitis how this is very marked, very labeled. And this is in the brain, the post-mortem brain of people with Alzheimer's. And you can see here that there are some specific places where you have a lot of this gingipine. When you get the image a bit bigger, you see here how the typical shape of Porphyromonas. So you can have either the whole cells arriving there or the toxins that they produce. And to make things even more interesting, it seems that the place where the gingipine appear to be located is the same of where the amyloid plaques appear to be developed. And you know that amyloid plaques are the ones that actually are going to be responsible for the disease. And there are several research teams that have shown that amyloid has antimicrobial properties, and they believe that actually they could be an immune response against invasion by pathogens, including these toxins from Porphyromonas. So I think that this is very interesting, that should be more studied in the future. And it really, we have to tell our patients and also the people who are going to perform the public policies, protocols and so on. I think we all have to be aware, and society have to be aware of how important to keep a good oral health is, not only for oral health, but also for the systemic health. Of course, not all the effects of oral bacteria are going to be bad. I told you before that good oral bacteria in the oral cavity are able to transform nitrate into nitric oxide, and nitric oxide actually is an important vasodilator. So it has many positive properties in the human body. One of them is that it is a vasodilator, and therefore bacteria could have an influence on our blood pressure, as I'm going to show you. So the way we test this in the lab, we drink a glass of beetroot juice, which is rich in nitrate, and what happens is that our body has found a way to recycle this nitrate so it is not lost. Of course, how much time is the food going to be in the oral cavity in contact with the bacteria so they can do this job for us? Just for a few minutes, right? While we are chewing on the food. But this is when things get really interesting, because when we swallow it, the nitrate can go into the bloodstream, and our body has found a way to recycle it. So when the blood is close to the oral cavity, the salivary glands actively capture that nitrate and put it back into the saliva. And therefore, when we measure nitrate in saliva after having a drink of this beetroot juice, after half an hour, like magic, we have a peak in nitrate levels in saliva. Then we swallow it again, it goes back into your blood, and then again recycled into saliva. And then as a consequence of that, after having a vegetable-rich meal, your levels of nitrate stay high in saliva for more than 5 hours. So the body makes sure to feed our microbes, like that little factory of microbes, to to feed them the nitrate so they can produce this nitric oxide. Of course, when that happens, nitric oxide acts as a vasodilator. It's going to expand the blood vessels. It also improves endothelial function, and it has many positive functions in the body. That second step from nitrite to nitric oxide can also be performed by humans. So either chemically in the stomach, because at an acidic pH, this is chemically transformed, or because we have enzymes that are able to do this. This step can be done by humans, but that first step of transforming nitrate to nitrite can only be done by oral bacteria. Then if we measure the nitrite, NO2, we see that it takes a bit longer. So this is the nitrite levels in saliva. After half an hour, we have the peak in nitrate. After 1 hour, we have the peak in nitrite, and the nitrite levels remain high for a few hours, allowing the body to transform that into nitric oxide. Of course, if you have a mouthwash with an antiseptic, we kill temporarily the bacteria in the oral cavity. And you have to be aware that when we have a mouthwash with an antiseptic, we kill not only the good oral bacteria, but we also kill the pathogens. Sorry, so not only we kill the pathogens, but we also kill the good oral bacteria, and that means that they are not able to reduce the nitrate. So when we look at the nitrite levels, NO2, in a person that has had a mouthwash with an antiseptic, there are no nitrite at all. So I think that, of course, having a mouthwash is important when the dentist says that you have to use it for particular occasions, or you have a gum disease or under particular conditions. But in my opinion, it is no good to actually use a mouthwash with antiseptic on a daily basis as part of your daily oral hygiene routine, because then you are wiping off all the microbes, and we need to keep them for many positive functions, like, for example, nitrate reduction into nitric oxide. And it's very interesting because the proportion or the levels of these nitrate reducing bacteria are extremely low in people with gum disease. So when in periodontal patients, we have an increase in all these pathogens, an increase in the red complex bacteria and in other bacteria which are associated with gum disease, at the expense of a very strong decrease in bacteria able to reduce the nitrate. We have seen this in many countries. This is in Brazil, for example, an increase in pathogens in the red complex bacteria in people with periodontitis, and a huge decrease in nitrate reducing bacteria. So we believe that if you don't have these bacteria, then you cannot transform nitrate into nitric oxide. So we believe these people are going to have a deficit in nitric oxide. We have shown that this is the case. In people with gum disease, they have lower nitric oxide levels, and then they cannot benefit from the blood pressure lowering effects of the vegetables. And we actually think that the connection between the oral and systemic link cannot only be due to the inflammatory markers, which are produced by pathogens, but also by the lack of nitric oxide production by the health-associated bacteria. So I think that in addition to the standard, to the current idea that the connection between gingival health and systemic health is mediated by the pathogens, I think we have to consider also the deficit in nitric oxide, because people with gum disease are not able to reduce the nitrates. It's very interesting that when we treat, when the patients with gum disease are treated, then these nitrate-reducing bacteria come back to the oral cavity and they recover the ability to reduce nitrate and to produce nitric oxide. And therefore, we see an improvement also in blood pressure. So I think this is my final slide. I just want to say that in the past, the link between the oral health and the systemic health was mainly focused on periodontal pathogens, creating a systemic inflammation state. But I think now we have this extended view by which oral bacteria are not only important or not only have an effect when you have disease, but also under healthy conditions, we see that good, healthy oral bacteria are able to produce nitric oxide, and probably, we are testing whether they are also able to produce some other beneficial compounds. We have also found that they are able to produce essential amino acids that are able to contribute to our general health. And I think it will be very interesting to look at whether they are able also to produce some other compounds that could be important for our health. And some of these compounds will be released directly into the bloodstream, but we also believe that most of the effect, or a big part of the effect, can be mediated by the saliva, because you can produce all these beneficial compounds that go into the saliva, then you swallow the saliva, and then that's going to be absorbed and have an effect on the rest of the body. So this is what I wanted to share with you today. I want to thank all the people from my lab and all the collaborators in Sweden, in the UK, in Mexico, and in Spain at different institutions, as well as in the US for the different studies that I have shown you today. And thank you very much again for your attention and for the invitation. And I will be very happy to take any questions that you want to make. Thanks.
Alex, thank you so much for such a wonderful presentation. I am really impressed to see how many updates you are able to cover in this 1 hour. Really amazing. I'm happy to report that we will be able to, at the end, edit this presentation, cut the details and the beginning, but having it posted in our YouTube channel, I think is worthwhile to go back and review, because it's a lot of information, lots of new information. And I think especially for us that are not microbiologists, I think the way you addressed the topic was just fantastic. I want to thank you so much for all of your contributions here and the amount of scientific evidence you brought it to us today. I just want to tell the audience that you can type your questions in the question and answer box, and then that Dr. Rodrigues will be mediating the question and answer session. Thank you, Alex.
Thank you so much for your words. Thanks.
Alex, once more, an amazing presentation. Very, very thanks for that. We have on the Q&A a question from a special person, Beatriz Neves.
Hey, hello.
That had the opportunity to be with you in Valencia, learning with you. And she said, I want to ask about the association between Candida and Streptococcus mutans that you mentioned. We recently published a study where we performed PCR on caries dentin lesions in permanent teeth, but we did not identify Candida albicans and mutants together. However, many studies suggest that this association between Candida and S mutants in early childhood caries. What do you think that could be the hypothesis for this association potentially being more significant in children, especially related to early childhood caries?
Right, it's a very good question, and well, very, very happy to listen, to a question from Bea. I hope you're doing fine. Then, yeah, it's a very good question, and actually, I don't know. I don't think it is known why it appears to be more significant in early childhood caries, but not in adults, for example. So I really don't know what could be the environmental pressure. It might be something related to the immune system. It might be that the immune system in children is still immature, and perhaps Candida is able to invade more successfully. But I don't think it is actually known what is the underlying reason. It would be nice to know why it happens. It could also be related to different strains that appear to be at different proportions depending on the age, but I think that this is less likely, and I think it's probably something to do with the immune system that still has to be identified.
Thank you, Alex. And also these samples were related to dentin caries, and I guess that is probably more important in enamel caries. I don't know.
Right.
The other messages are of thanking and congratulations for your nice presentation. And we have another question from Nicole de Melo Fialhos. periodontal disease are caused by a dysregulated immune response against dysbiotic microbiome. Is there any evidence that probiotics can also lead to a more homeostatic immune response from the host?
Right, very good question. Yes, actually the action of the different modes of action of the probiotics are very interesting, because one of them is related to the inhibition of the pathogens. So you have lower levels of the pathogens and therefore the inflammation appears to improve. But also some of the surface proteins in the probiotics actually directly inhibit the production of inflammatory cytokines. We have, this has been shown with different bacteria. We have also shown that with S. dentisani. And it's very interesting because sometimes it still has this anti-inflammatory potential on gingival cells, even when the bacterium is dead. So you expose the gingival cells to the live and the dead organism. And when the organism is dead, it still lowers the amount of these inflammatory molecules. So it must be some surface proteins that actually has this effect. There is also another way by which you could improve the immune impairment induced by oral pathogens, because some streptococci, for example, on their surface, they have small peptides like this 11 amino acid peptide present in some streptococci that specifically inhibits the virulence factors of Porphyromonas gingivalis. So it is known that it doesn't kill the organism, but it inhibits, for example, gingipine expression. So I think, again, we are here, to the level of how the interaction between different bacteria have an effect. For example, gingipine is produced by Porphyromonas gingivalis, which is the pathogen, but we know that the presence of another organism, which is Parvimonas micra, actually activates or induces the expression of gingipine. So perhaps this second organism, we cannot consider it a pathogen, but it needs to be present for the pathogen to secrete the toxin. And then we have some health-associated streptococci that have a peptide that inhibits the expression of the gingipine. So yeah, so I think there are different ways by which probiotic organisms or health-associated bacteria can influence the immune system, by either killing the organism, the pathogens themselves, by inducing or repressing the immune response from the cells, or by inhibiting the production of toxins that are going to have an effect on the immune system.
Thank you, Alex. And we had another question from Pedro Ricomini from Unicamp, Brazil. Thank you very much, Alex. How could quorum sensing molecules be useful for controlling biofilms related to oral diseases?
Right. Well, yeah, I think the only use so far that we have seen in the oral cavity is the reduction in the biofilm mass. We know that the amount of biofilm is very important for oral diseases. So, for example, the more biofilm mass that we have, the more dental plaque accumulation, the more caries, because the pH drop is larger. There classic experiments in the 80s and the 90s where you can have a look at the Stefan curve. When you brush your teeth, so you have a very small amount of dental plaque. Then you wash the oral cavity with a sugar solution. Then you have a drop in pH, which is very small. If you don't brush your teeth for 24 hours, then you have the mouthwash with sugar, and then the pH drop is much stronger. It happens faster, and it takes longer for the pH to go back to neutral. And if you don't brush your teeth for 6 days, then it takes a long time. So I think the more dental plaque, more caries. The more dental plaque, more inflammation as well. So we know that, a bad oral hygiene, you accumulate dental plaque, that is going to induce inflammation. The body reacts by the production of the gingival fluid, which is very rich in, it's a serum, which is very rich in proteins. And then these highly inflammophilic proteolytic organisms, which are responsible for gum disease, they increase in proportion. They create more inflammation, and then you have a vicious circle. So, and the same is also true for halitosis. You have the biofilm on the tongue, dorsum, and the thicker the biofilm is on the tongue, the higher production of volatile compounds involved in bad breath. Therefore, any strategy to reduce biofilm mass is going to be positive for oral diseases. So that's why one of the signals, that bacteria use to decide if they form a biofilm, if they produce this extracellular matrix, is actually the density of bacteria. And how do they know that they have many bacteria? Because of the quorum sensing signals. So by blocking those signals without killing the bacteria, we are using their own weapons. We are impeding them to communicate with each other so they don't decide to form a biofilm. I think it's a very clever strategy. In other diseases outside the oral cavity, the inhibition of quorum sensing is being employed for other reasons. For example, because in some bacteria, they use quorum sensing to produce toxins. But as far as I know, there are no cases in the oral cavity where the production of toxins induced by quorum sensing has been studied to be inhibited by quorum quenching molecules, but it could also be interesting.
Thank you. Regarding quorum sensing, you mentioned bacteria, marine bacteria or marine organisms.
Yeah.
It's easy to find this organism.
Right. Well, there's a system by which in culture you can identify, you have a reporter, you have an organism that forms quorum sensing that produces quorum sensing, and then it changes color. And if you actually happen to find a molecule that is going to bind and to block this, then you stop seeing that color. So this is the way that this group in Santiago found this marine organism able to produce this molecule. They identified the molecule, and then that's the one that they have used. And I think they've been very lucky, because when they applied that in the oral cavity, the quorum quenching molecule, this quorum sensing inhibitor that they collected, happened to be of the same family of quorum sensing molecules, which is very common in the oral cavity. So it worked by chance, but it worked very well. But actually then what we did was to do a deep analysis of all the quorum sensing molecules present in the DNA of the bacteria from the oral cavity, and we found a lot. I just couldn't believe that there are so many quorum sensing molecules and quorum sensing inhibitors encoded in the bacteria. So I think we just have to look for them and try to apply them. So, because I think that if using a quorum sensing inhibitor that comes from a marine bacterium, it works, I think we can even improve this further by using quorum sensing molecules from the oral cavity. And I encourage the audience to actually do research in that. I think it's a very promising strategy.
Once more, thank you, Alex. We learned a lot with you. Sorry, Adriana.
I cannot type in the box because I am a panelist, so maybe I can just go with one question, because I know we are over time already, but it's such an interesting topic. I'm really, excited and happy to see your discovery in the Streptococcus dentisani, and now how you leverage your discovery towards development of a material that can be applied on patients' mouth and control disease. So when you first found that, Petri dish with that huge halo around the dentisani, I am sure you further investigate. What is the mechanism? Is quorum sensing? Is inhibit attachment? What is the mechanism involved in the dentisani to create that huge halo or inhibit? And you mentioned about the nitrate mechanism and so on. But then an additional question attached to it, since you are applying the patient's mouth and the patients, for sure, you have a mucoadhesive, as you mentioned, but this material will be at the end swallowed by the patient. So what is the concentration of the bacteria, and how do you envision those dentisani in the gut microbiome?
Okay. Yeah, very good questions. It has been a very exciting period. You know, when we first discovered it was 10 years ago. So this is the period that has taken us from the discovery in the lab to doing the scale-up production, all the safety in animals, the clinical studies. You know, it has really been a very long path, but also very excited. Although I have to say that most of the hair I have lost and most of the gray hair I have is actually for the development of Dentistry as a probiotic. But so in relation to the dose, what we did was, okay, let's look at the levels of S-dentisani in healthy individuals. We saw that individuals with dental caries that have very low levels, some individuals have 0 and others have a little bit. Individuals who are caries free, they have much higher levels. And in those individuals, we quantified and they have about 10 to the 8 cells in the mouth. So we said, okay, we are going to apply 10 times more because as you're saying, when you apply it, a big part of it is you are going to swallow it, it's not going to grow. So we decided to give it 10 times more. So the dose we are applying is 10 to the 9 cells per dose. Then as part of the safety issues that we have to study was whether those bacteria are able to go to the gut. So we first did that in an artificial stomach model, and S-dentisani dies after 5 minutes in the stomach. So even though we swallow it, it cannot reach the gut. This is just the opposite of what you want with a gut probiotic, that you want to survive the stomach, so it goes into the gut, you know. And when we did these studies, you know, we did this in an outside company, and they were very sad, saying, you know what, they all die. I'm very sorry because it doesn't reach the gut. And we were like, no, this is good for us, you know. We don't want it to reach the gut. So and then yeah, we apply it in this gel. We have to give it some time for the bacteria to regrow and to be able to attach. And you also asked about the way it works. Yeah, so at the beginning, we thought that it was able to produce one of these inhibitors. And we were surprised when we examined the genome and we saw that it had 14 of these antimicrobial peptides. This is the highest number of antimicrobial peptides present in any bacteria which is known in nature. Is the one with the highest. We were very surprised. So we thought, okay, then it's not able to kill only S mutants. So we started to see, and they appear to have very little effect on many of the commensals, and they have a larger effect on many of the pathogens. So it kills Fusobacterium, for example, which not only would be good for dental plaque, but also it would inhibit the bacterium that we know, you know, can cause colon cancer. It inhibits Porphyromonas gingivalis, Tartanella forsythia, Prevotella. So it's actually active against many, I mean, to different extent, but it's active against many pathogens. What we are doing now is we have ordered the synthesis, the chemical synthesis of each of those 14, and then we want to know each of them against which bacteria is active. And the idea is that, okay, we can have the Dentisani in a toothpaste, you know, but we can also put a combination, a cocktail of these antibacterial peptides with specific uses. So if you want to have a toothpaste against halitosis, you know, then we are going to put only the antibacterial peptides there that are active against halitosis pathogens, or to have a specialized toothpaste with people with gum disease. So that's another application. And so far, it's taking us some time because some of these antimicrobial peptides appear, you need to have 2 of them to work. This is also something common in these kind of peptides. So in some cases, you only need 1, in others, you need 2. And then we are trying to see how can this be also applied to mouthwashes or toothpaste.
Wow, this is amazing, Alex. Really amazing work, and we can really see how busy you are and how much work you
yes.
have. But you know, you are bringing such a promising area of research that goes in a different than what we have done until this point. So congratulations on all the work you have done, and thank you very much for being with us today. I think as the audience and the other panelists, we really enjoy your presentation a lot. If you want to give any final remarks, also Dr. Rodrigues, if you want to give any final remarks before we end the session.
Thank you very much for your words. I hope I have been able to transmit, the passion that we are seeing. I think that the last 10 years has been absolutely wonderful. The field is really changing a lot, and I can really see that in the near future we are going to see many changes in the profession. And I really hope that in the future, in the dental clinic, we will do more work in relation to the prevention, in relation to the diagnostics, and also, one of my goals in the rest of my career is going to be able to put again dentistry at the center of human health. I think that in many countries, when dentistry in the degree, was separated from medicine, I think in a way that implied that many medical doctors don't know much about the oral cavity. Sometimes when I talk to them, I have the feeling with some of them that they think that the human body starts from here down, and so I think, we have to be able to transmit this to our patients, how important oral health is for the general health, and also to the medical doctors and to the policymakers as well. We are seeing, for example, that most of the pathogens are able to colonize the baby at the very first year of life, So if the mother or the people who are in close contact with it actually has a bad oral health, many of these oral pathogens are going to be exposed to the baby, and the immune system of the baby, that when the baby is born is very naive, that naive immune system is going to generate immune tolerance against those pathogens. It's going to consider that they are good bacteria, and therefore it's going to be easier, that the immune system is going to tolerate them, and it's a very bad start. So if we are able to, with the evidence we have now, we are able to talk to the politicians and to the policymakers and say, look, if we give free oral care for pregnant women and women during the first year of age, that is going to save so much money and so many diseases in the future for those children, that we need to take this into consideration. For example, this is just one example about how scientific evidence that we are getting now can be used to improve our lives. And I think we are in a very exciting moment. And I have also seen that there's more and more research being done in the field. You just have to look at the impact factor of the journals in oral sciences in the last 10 years. They have increased a lot because there's more work about it. And I think we are at a very exciting moment to be doing this. Many people studying the dentistry initially go there because of the clinical applications. And I think now there are more and more dentists that are also being involved with research. And I think we are all part of the same chain, On one side, we have the scientists, we are doing research that later on will be applied in the dental clinic. And I think we have to communicate more with each other, as we are part of the same chain and we all want the same,
Exactly what we are trying to do with the cluster, to make it an interdisciplinary, multidisciplinary group of people with mind like it.
Thank you. I appreciate it.
Alex, I have only 2 words to talk. The first one is thank you again. And the second one is congratulations. It was an amazing presentation, and also congratulations by the choose of the name of the 14 peptides, the new peptides, ACDC.
The dentistry.
Thank you. Thank you.
You're welcome.
Thank you, everyone, for staying with us until now. The session is now ended.